Hello everyone, myself Praveen Baliram Roylawar. Today I am here to present my research work entitled Physiological and Molecular Characterization of P. indica priming against onion stem phylum Vesicarium interaction. This work has been carried out in collaboration with ICIRTLS, that is Indian Council of Agriculture Research, and Director of Onion and Garlic Research, Rajgurunagar, Pune. So the funding for the research has been availed from the DOGR itself. So we'll be very thankful for the DOGR funding. So as we know, so onion is a very economically important crops. So it is, and India is topmost producer of onion, second topmost producer of onion all over the world. But what is so different in this topic is that this study has not been carried out so far in onion. Means this is very the first time we are using this P indica, which is an endophyte, in order to generate resistance in the onion plant. That is, in order to generate induced systematic resistance in onion plant against a crucial fungal pathogen like stem phylum Vesicarium. This is the novelty of this study. So, as we know, though we are largest second producer of the onion, but yield loss is the very first factor that we came across when you thinking of the when you think of the increase in the yield. And there are many factors like biotic and abiotic factors are responsible for the yield losses, but the biotic factors like diseases are the most important. Among the diseases, I would like to add here the fungal disease. This is not due to fungal pathogen is a major is a prime important where the researcher should focus while considering the onion plants. So for overcome the loss due to the fungal pathogen, we generally use a lot of chemicals in the fields like fungicides. Farmers use a lot of different fungicides in the field in order to overcome such disease. But as we know, fungicides are very harmful for the human as well as environment. So our motto is to reduce the fungicide spray in the field as well as to increase the plant vigor and to protect the plant from the pathogens. So we have in this study we have included root endophytes like Piriformospora indica is a known endophyte which has been used in many crops like many we can say it has been used in many modern crops all over the world, but this particular fungus to our surprise has been never tried in the onion. So what we have done, so I would like to add here, I'm not going much more details in material and method as due to the time limitations. I'm mostly focusing on the result sections, and while explaining the result sections, that's what I included in my slides. I will brief the material and method as well. So what we have done, so first the question arises, is this fungus really works in the onion? So for our study, we have selected one stem phylum Vesicarium susceptible variety. On that variety, we first colonize the plant with the Piriformospora indica. Here we can say Pi plus means colonized plant and Pi minus means uncolonized plant. Okay, so on this thing we would like to test the plant vigor. Is onion plant is responding to this colonization or not? So after 60 days post colonization, first we have to check whether it is fungus itself colonizing the roots or not. So what we have said, after 60 days post colonization, we have checked the colonization process. So we came to know, yes, this particular fungus, that is this particular endophyte P. indica, is actively colonizing the roots. This is what we have checked with the normal staining process, okay, microscopic process. Then further we want to test it on molecular level as well, whether it is colonizing or not. So we have used the P. indica specific primer, that is P. indica transcription elongation factor. This particular primers we have tested. We have extracted DNA from the roots of colonized and non-colonized plants, and we have amplified this particular DNA with P. indica specific primer. Then we came to know, we can see over here in P. indica colonized plant, we can see the bands, that is the bands of PITF genes. That is this gene is from the P. indica itself, and in uncolonized plant we don't see any band. So on this Bros process, like on in microscopic process and on molecular level, we have confirmed the colonization of this particular fungus, means this particular fungus is colonized the plant. But when this particular fungus is colonized, we have also seen that there is tremendous increase in the plant vigor. As we have just communicated this paper, throughout the presentation, I'm highlighting the important data. Okay, I'm hiding the important data that I don't want to disclose, and I don't have authority before the publication to disclose this data. So in late stage of infection, in very few data I'm disclosing over here. So please cooperate with this. So we have checked the leaf length, root fresh weight, and root dry weight in control and P indica colonized plant, and we came to know that okay, we have find out that okay, P indica is showing very significant changes on all these aspects like root length, leaf, length, root fresh weight, and root dry weight. P indica is showing very significant changes. Here in the picture, you can see the colonized plant showing very active growth. Okay, active vigor and growth. In unique root, we can see we can easily make out the difference in uncolonized and colonized plant, as the number of roots and root lengths are typical has changed significantly. After this, we have went further. Then what we have done, as we wanted to test, okay, the plant vigor is increasing, but whether it is giving resistance against the pathogen, that we would like to check. Then these inoculated plants, that is after 60 days post transplanting, okay, all this experience is going in greenhouse at this stage. We have inoculated the plants with the stem phyllium vesicarium spores. uh, then we are after inoculating, we have tried to see the symptoms, okay, which is appearing on the inoculated plants and non-inoculated plants. So we have seen such symptoms in the greenhouse experiments, and these symptoms we have noted it down. We have calculated the percentage disease index, whether the percentage disease index in inoculated plants is high or less as compared to the control plants. So here you can see the PDI, in the non-inoculated plant, that is water-treated pathogen inoculated. W plus P stand for water-treated pathogen inoculated plants. The PDI for in this particular plants is very high and is progressively increasing. We calculated PDI at 135 day post inoculation. Means after pathogen inoculation, we have waited for 24 hours. Like that is 1 DPI. Then after 3 DPI, we have taken the reading. After 5 DPI, we have taken the readings. So we came to know that in water treated pathogen inoculated plants, that is controlled pathogen inoculated plants, the disease is progressing very high. Then we came to know that okay, but in P indica colonized pathogen inoculated plant, that is P plus P, where we colonize the plant with the P indica, the pathogen progress is significantly reduced. It is very significantly reduced. Then we are asked our question, what is the reason behind it, why the disease progress is reduced? Then we know when the stem phylum vesicarium is a necrotrophic pathogen. So in order to make the disease, it has to kill the cell, and many times necrotrophic pathogen use the mechanism of generating the hydrogen peroxide or oxidants in the leaves that affect the cells, that kill the cell. So we went for the level of hydrogen peroxide in the colonized and uncolonized plants. So I'm focusing only in the colonized and uncolonized plants here. Okay. So in colonized plants, we have seen the level of hydrogen peroxide is too much as compared to in uncolonized plants, the level of hydrogen peroxide is very much as compared to the P indica colonized plants. So there are two ways the hydrogen peroxide is works. Okay, if its level is tremendously increasing, then it is very toxic to the cell itself. But if the level of H2O2 is balanced, then it acts as a signaling molecule as well. Okay, so here first we have find out, yes, hydrogen peroxide is level of hydrogen peroxide is too much in the uncolonized pathogen inoculated plants. Then we said, okay, the level of hydrogen peroxide is increasing, whether it is causing the damage to the cell or not. For the cell damage, we have done the lipid peroxidation, and we have measured in terms of monoaldehyde dehydrogen MDA content. We have measured in terms of monoaldehyde context. So we have seen the damage as the level of hydrogen peroxide is increasing uncolonized plant. that it is also causing the damage to the cell itself in uncolonized plant. So you can see here the level of damage is increasing, lipid peroxidation is increasing, but in P. indica colonized plant, the damage to the cell is very less. Yeah. So we have come to know that yes, the hydrogen peroxide is increasing and it is also causing damage to the cell in uncolonized plant. On others, in other cells, it is not causing damage to the P. indica colonized plants. So there is a mechanism in plant to counteract or to nullify this oxidant generation. They try to detoxify this. We do check it by with the help of antioxidant defense mechanism. So we have checked 4 antioxidant defense mechanisms like catalase, ascorbate peroxidase, peroxidase, and superoxide dismutase. And we also check one defense enzyme, that is phenylammonia lyase as well. So what we came to know here, that in P. indica colonized plant, P. indica colonized pathogen inoculated plants, we can see that catalase, superoxide dismutase, and peroxidase. These are the 3 enzymes, okay, which are significantly high. Like catalase is playing high at the initial stage of infection, like at 1 dpi. Yesterday also is very significant at 1 dpi, but peroxidase is significant at all stages of infection. While what we can see in uncolonized pathogen inoculated plant, that is W plus P plant, we can see here ascorbate, the level of APX, ascorbate peroxidase, is high in initial stage of infection, that is 1 dpi and 3 dpi. 1 dpi and 3 dpi. And when we came across the phenylammonia lyase, which is responsible for generation of salicylic acid, it is play key role in salicylic acid pathway. So we can see here that phenylammonia lyase is very significantly high in pathogen controlled pathogen inoculated plants. So what we make the story out of it, like here the modulation of this oxidants, modulation of this enzyme is taking place, where P. indica colonized, where we have colonized the plant with P. indica and inoculated with the pathogen. So we can see the modulation of antioxidant defense enzyme, because where some enzymes playing in initial stage of infection, and some enzymes playing throughout level of infection we have tested. So for in order to suppress the disease, the modulation of this enzyme is very important, and this what we have seen in case of P. indica. So what we came to know, okay, we are not stopped over here itself. We also know that some hormones play a very crucial role in stopping the disease when we consider necrotropic pathogen. As we there are many literature stating that necrotropic pathogen use the salicylic acid pathway in order to progress the disease in the plant itself. So we have tested 2 hormones like salicylic acid and jasminic acid pathway in water treated pathogen inoculated plants, that is controlled pathogen inoculated plants, and P. indica colonized plant as well. So here we came to know the reason beyond generation of H2O2 and high level of H2O2, high level of peroxidation, could be the level of salicylic acid, which is tremendously increased in the pathogen inoculated plants. This hormonal analysis is done in late stage of infection, that is at 5 dpi. So we kept note at 5 dpi in water treated pathogen inoculated plants, the level of salicylic acid is very high, while in P. indica colonized plant, the jasminic acid level of jasminic acid is very high. So as we know, in order to counterattack on the necrotropic style of pathogen, plant use the jasminic acid pathway. That many literature has been stated that J is a very jasminic acid and its responsible genes are very play a crucial role against the necrotropic style of pathogens. So we came to know that okay, here SA level is increasing in controlled pathogen plants, and J level is increasing and in P. indica colonized plant. This study is outsourced from the Genome Bio company. They have done this analysis with the GCMS technique. So as we know, okay, SA and J is playing role. We further know that SA and J, they also regulate some sort of genes. Okay, they play a crucial role in upregulation or downregulation of genes, because these are defense hormones. We term these hormones as a defense hormones. So we went further for on the molecular level. We have taken together 8 genes for the this analysis to in order to explore, in order to characterize which particular genes are playing crucial role in plant defense, means in this particular onion stem phylum interactions. So we want to check that. So what we came to know, this LOX1, LOX2 and AC chitinase. LOX1 stands for lipoxygenase. Lipoxygenase 1, lipoxygenase 2 and chitinase. These three particular genes are highly upregulated in P. indica colonized plants. And as would like to add here, lipoxygenase LOX1 and LOX2 are the marker gene for jasminic acid pathway. Means, as we know, jasminic acid is increasing P. indica colonized plant, and the same genes jasminic level is increasing, and this jasminic acid level is controlling this gene as well. But and when we check in water treated pathogen inoculated plants or controlled pathogen inoculated plants, what we came to know, that okay, there ACGST, PAL1 and WRKI70, these genes are upregulated. ACGST stand for Alimsipa glutathione S transferase, and this is the gene is a marker for salicylic acid marker for salicylic acid. Salicylic acid. So we keep, and PAL1, as I told, it is also playing level of salicylic acid, and WRKI70 is a transcription factor. There are so many transcription factors, and as we know, WRKI70 is playing it is get upregulated in response to the salicylic acid, and further it regulates further genes like PR1, like pathogen related protein genes, it regulates. So what we the novelty of this study is that the genome sequences of anemia is not available. So far, very few paper, you can count on the palm, like you can count in number 2, 3, 4, only 2, 3 papers are available where the study of the molecular levels has been studied. And but this is our first study. We have included 8 genes, irrespective of the genome sequence of alien is not responsible. We have scanned through the databases. We have found out these genes. Okay, these genes we have included in our study. Very first time these genes are included in the study itself. So we came to know that okay, not only the level of jasminic acid is increasing, but also the level of but also the marker gene for the jasminic acid is also getting upregulated. Then we have make the statement like okay, here could be the mechanism of P. indica to transfer the resistance to the onion plant against the stem phylum basicarium could be the way by use of jasminic acid pathway. Jasminic acid pathway might be the playing role here. Then we have went for the field study itself. In field, this is the I would like to add over here, sir. P. indica is a globally studied endophyte, but very few study has been carried out in the field itself. And this is our we have tried to attempt this in the field because ICIRTLS itself believes in the transferring the technology in the field, transferring, making it applicable in the field as farmers should be get benefited. This is our main aim of this particular institute and our goal itself. So what we have done in field, we have not only included untreated and P. indica control and P. indica treatment. We have also included the fungicide which generally farmers use in the field in order to protect the plant from the leaf blight pathogen or other fungal pathogen as well. So we have included 3 fungicides, that is Abriotop, Amistrotop, Propiconazole. That Amistrotop is a systemic fungicide like Abriotop as well. So these 3 fungicides we have included on the field. And what we came to know, and this study we have carried out in 2 years of Kharif season, like 2018-2019 Kharif season, we have carried out this particular field experiment. We have find out that the control plants showing the high level of percentage disease index. We have carried out this is in PDI. We have recorded PDI value at 3 days post transplantation, 3 days, 45 days, 60 days, and 75 days post transplantation. This is generally where the leaf stage of the onion is very much important. As long as the leaf stage is very healthy, leafy stage is very healthy, it will benefit the bulbs as well. So that's why we have carried out at 30, 45, 60, and 75 days day post transplantation of the onion plants in the field. So we have inoculated P. indica to the onion plants with the dipping methods. I won't go much in detail due to time limitation, but we can what we have find out that in control plants showing high level of PDI, but as compared to control, P indica plant has shown very less PDI, that is very significant reduction in the percentage disease index. But when we compare, as you can see, when we have compared the fungicide, all the fungicides showing very prominent result as to control the leaf blight in the field, like cabriotop, amistrop, propiconazole, all are significant as compared to the P indica. But among the fungicide itself, amistrop is the fungicide has shown the consistent and very significant results controlling this particular leaf blight. Then we can ask the question, then if the fungicides are very useful, then why to go for the biocontrol agents? But as I said, we want to minimize the spray, minimize the minimum use of chemicals in the field itself, because the fungicides, it costs a lot of money to the farmer. Then we have proposed, like now we are doing further studies, like okay, we would like to we have proposed that okay, by using P indica and this amister top, combining both these, at least we may minimize the spray of fungicides in the field. Our study also going in that direction, that is the further project we are carrying it out. So what we have concluded, it is an efficient endophyte which is increasing onion growth and defense against the fungal pathogens like stem phyllium vesicarium. Then the which mechanism? So it is balancing the generation of H2O2, as we say, very less H2O2 generated in the plant with by efficient scavenging of the H2O2 through POD and APX, because we have to manage the level of H2O2. How we manage the to detoxify, make it making what antioxidant do? They make H2O2, they convert H2O2 to water itself to detoxify. So they are uh, so they are controlling the level of H2O2 here. So we also seen the prime plant show the enhanced expression of chitinase, a pathogenesis related protein gene, which could have played a key role in restricting plant pathogen colonization. As you know, chitinase act against the fungal pathogen by dissolving the chitin in the fungal cell wall. So this is what we have concluded. So we are actively working groups. So this is our publication from 2019. We have published in very reputed journals like PLOS One, Plant Disease. We also came with the research articles like the approaches in the allium research. So we are actively working over there. So I'm very much thankful for the organization to give me this opportunity to publish to present my work, and I'm very thankful for our group. So they are giving the very best of them, and I'm very thankful for the our principal scientist Suresh Gawande sir. He is the main team leader who actually controlling the funding for the works as well, and we also is getting guidance from the BP Shinde sir, Bharat Shinde, and Gawande sir. There are other colleagues as well. So we are team of people from the different backgrounds. Like some are bioinformaticians, some are uh, some are desire, some are handling the field works as well. So I'm very much thankful for group to let me allow to present these works. So we have communicated this work in the Elsewhere Biological Control with impact factor 2.754. This paper is under still review. For this purpose, I have hide the I hide the data in this presentation. So thank you very much for this opportunity.